All right, we are counting on you remembering mitosis from your earlier years in biology, and if you don't, this is your chance for review, okay? To make more cells, we have to divide cells. But before we divide, we've got to replicate that DNA, remember. This is the part, however, of the cell cycle. We're not going to talk too much about DNA replication. What we're going to talk about is this tiny little sliver of the cell cycle called mitosis, and we will also talk about a little bit about cytokinesis, but that's not going to make up the bulk of this. So we've already talked about why cells divide by mitosis. We want clones, if you'll recall. Now there are some components that we are going to focus on. In mitosis, we are focusing on the nucleus, specifically the division of the replicated chromosomes, the replicated DNA, and that nuclear membrane. And we're also going to talk about the cytoskeleton, which is something we have not talked about a whole lot. But the cytoskeleton are the support structures and the structures that move things around in a cell. And in order to divide the cell, lots of stuff has to move around. The chromosomes have to move around. We have to cut the cell in half, and it's the cytoskeleton that's going to be involved in that. So that's going to be an important part of our focus. So let me introduce you to a few cytoskeletal terms and concepts. The function of the cytoskeleton is support. Skeleton, cell skeleton. It's kind of like your skeleton. It's there to support, help the cell maintain its shape. It's made out of protein, as are most things in your body. The cytoskeleton is made out of protein. There are three types of fibers. The ones that we will be talking about are microfilaments, and perhaps most importantly of all, microtubules. So those are all parts of the cytoskeleton. These guys, intermediate filaments, really don't do much. They really truly are kind of just scaffolding, but the other two actually do a lot of activities. Things like motility, like allowing the cell itself to move. Cilia and flagella are made out of microtubules and allow cells to move. The organelles inside cells are allowed to move also because of microtubules. The cell will be able to cut itself in half because of microfilaments. Your muscles work due to microfilaments. So those guys are really important. We are focusing, though, a lot on microtubules this time. That's just showing you some a real cheesy diagram of some cytoskeletal components that might be in a cell. Now, centrioles are really important. These are little organelles that exist only in animal cells. Plant cells don't seem to have them. But the role of the centrioles is to organize the microtubules correctly so that they can attach to the chromosomes and pull them apart. They make what are called spindle fibers, or they organize the spindle fibers. So what we see here, these are what the centrioles roughly look like. They exist in pairs, and during cell division, the pair splits up, and one goes to one side and one goes to the other side, and they organize these spindle fibers, these microtubules, and so they can attach to the chromosomes and ultimately pull them apart. So, what do we need to get to daughter cells? We need to get exact copies of genetic material, okay, which we will do by dividing that nucleus and the chromosomes exactly, but we also need to get some organelles to each of the daughters, some cytoplasm to each of the daughters, some cell membrane to each of the daughters, and so on, and cytokinesis is going to kind of take care of all that for us, okay? So, mitosis. Please remember we're talking 4 phases primarily: prophase, metaphase, anaphase, telophase, PMAT. Okay. Now, this here is showing you interphase right here. There's the centrioles being shown there. The chromatin, the undistinguished chromatin, that's interphase, where the cell would spend most of its life. But then remember those famous prophase, metaphase, anaphase, telophase. Be careful. Some books are introducing a kind of something in between prophase and metaphase called pro-metaphase, which just really seems stupid to me, but whatever. PMAT. Just remember PMAT. Okay? And then cytokinesis is the splitting of the cell itself. All of this stuff is dealing with just organizing and splitting the chromosomes. Okay? Let's briefly remember what's happening in prophase. The chromosomes get short and thick, and this says chromatin condenses. That's what that means. They get short and thick, and they become visible to us in a microscope. Okay? The centrioles that I mentioned are going to move to opposite poles of the cell so they can get in their position to organize the spindle. And the protein microtubules that make up the spindle are going to get moving as well. Okay? I'm going to cross this out right there because that's not true. The microtubules are made out of something called tubulin. Okay? At any rate, they're going to move the chromosomes, okay? And the nucleolus, which is really just a really active area of the chromosomes where ribosomes are made, that goes away. It's just a circular spot in the nucleus and it disappears. Also, the nuclear membrane is going to break down and go away, so these chromosomes can kind of spread themselves out and get organized a bit. So, it still looks pretty disorganized, but we've got a lot of activity happening in prophase. Okay? Now, this pro-metaphase kind of thing they've got going on now, basically this is kind of used to all be lumped into prophase. But the spindle fibers will actually attach to the centromeres, that region that is connecting the two identical chromatids. There are little spikes of protein coming off called kinetochores, and the spindle fibers are going to attach to those so that the chromosomes can be moved around. Okay? Just kinetochore proteins, kind of like handles on the centromere. Now, during metaphase, the spindle fibers are going to align each centromere of each chromosome along this kind of imaginary line across the middle of the cell, okay? And that is so they can be organized and split ultimately very carefully and very accurately, okay? So the spindle fibers are coordinating that movement, okay? Because we want to make sure that each daughter cell gets one of these chromatids each, okay? All right. Next is, this is just showing you another picture of that. Centrioles, the aster is just kind of microtubules around the centrioles, your spindle fibers, all that I think we're pretty good with. Anaphase, we are actually going to separate those chromatids, and they're going to move to opposite poles of the cell as the spindle fibers pull them apart. They actually happens because little motor proteins are going to actually walk these chromosomes along the spindle. Again, it's tubulin, not that. And ATP, of course, would be involved in this. It's a very energetic process. Okay? So we're splitting the chromatids. This is the important thing to remember right there. Okay? Now, we've already said all this. We're going from this double-stranded to this single-stranded state once again, which is what we started as single-stranded chromosomes. So we're going back to that, only we're going to end up having two cells. And we don't need this information. All right, telophase is when now we've got the chromosomes separated, and they are going to be at opposite ends of the cell, and this nucleus is going to start to come back, and things are going to start getting back to normal, but we haven't completely divided the cell yet. Okay, we have not completely divided the cell yet, but the daughter nuclei are reforming, the nucleolus is coming back, the chromosomes are going to get thin and stringy again, they're not going to be these short, thick things that we can easily see anymore, and cytokinesis, or the splitting of the cytoplasm, does begin, the actual division of the cell. During cytokinesis in animals, the actin microfilaments are going to be responsible. Remember, this is microfilaments, not microtubules. They are going to constrict, almost like somebody has a belt around the middle of a balloon, and we're just going to constrict this thing until it pinches inward. In a plant cell, oh, by the way, that's called a cleavage furrow, if you should ever see that written anywhere. And so in plants, if I can ever get to the plants, it's going to be different. Since plants have cell walls, there's going to be the laying down of new cell wall. There is no pinching in. We're just going to lay down new cell wall, and that's called forming a cell plate between two plant cells. So no pinching in, no cleavage furrow. Okay? All right. There, we're just seeing that. And I think... now, evolution of mitosis is thought likely to have evolved from the process of binary fission in bacteria. In bacteria, binary fission is not mitosis because there's no nucleus. It's just we're going to copy the DNA and then we're going to split the cell in half. The chromosome is circular. It's quite different, but it is thought that this is the roots of mitosis. And many of the proteins and things used in mitosis are quite universal to all animals and plants as well. So it's pretty ancient process for eukaryotes. All right, that's it. We'll catch you in the next podcast. Take it easy.